So we finally got through some of the more intense physiological aspects of this A&P1 course in the last sets of videos. And now we're going to apply that knowledge that we've had. What is the purpose of obtaining contractions? What are we trying to do? Well, we're trying to develop tension in muscles, right? To perform lifting, to move a load, and so, or just to move ourselves or to maintain posture. And so there are some aspects here that we really want to keep in mind. And we want to know that tension is produced by muscle fibers, right? So we know that we're going to have some internal tension, and external tension is exerted by the muscle fibers on these elastic extracellular fibers. So we're talking about translating the tension developed by muscles into the tendons, and then having the tendons interact with the bones in order to cause movement, change of position, postural changes, and so forth. What do we need to do to accomplish this will then result in a specific number of muscle fibers being stimulated. So in order to have our muscles functioning at a premium, at an optimal rate, right, and being able to conduct themselves at the maximum efficiency, we need to think about what kind of state they are in. And so we've talked about the sarcomere quite a bit, and we've discussed the zone of overlap. And what we're looking at now is maintaining a great relationship between the thin and thick filaments. We're going to idealize that zone of overlap, that distance there. We want it to be just perfect, because if it's just perfect, we're going to be able to maximize what our muscles can do. And so in this particular chart, we're seeing a variable that's called the optimal resting length, and we want that to be very specific. It is the optimal length that will give us the maximum amount of contraction. If it isn't optimal, then we're not going to get the most out of our muscles. And so you can see here in this particular part, this little bar here where this darker blue is the optimum resting length. It's not a huge area. So there's got to be a lot of work done and constant adjustment done through activity of the brain, translating into neuronal output and the development of electrical signals called action potentials, to get the muscle to the correct resting length so we can always have it at its maximal function. It's possible to have the sarcomere being overly contracted, and so that means we can't decrease the size of the sarcomere very much, right? If it's overly contracted. And that's on this side of the graph. So if it's overly contracted, as we move further away, well, that means the less distance that we can squish those thin filaments together. On the other hand, if we're overly stretched, you can see there's barely any bulbous heads of the thick filament that are overlapping the active sites on the thin filament strands in the actins, right? So there's not much opportunity for grabbing hold of those active sites and being able to exert a maximal contraction. And so you can see that on this right-hand side of the graph, how that affects the function of the muscle. So it's best to have it right here in the middle, and you're going to have that constantly occurring in your muscles, especially, let's take for instance your postural muscles, constantly adjusting them throughout the day for maximal effect and to maintain posture, because to do so also minimizes fatigue as well. So the maximum tension that we're able to develop on a muscle is dependent on primarily the rate at which those electrical stimuli are going to arrive at the muscle, and what our stretch is. What is our resting length, or what is our length of our sarcomere? Is it scrunched up and already partially contracted? Is it stretched so far apart that we can barely start a contraction? Where is that sarcomere at? How much oxygen do we have available? How much myoglobin is in the area to supply the oxygen? And how much glycogen, that storage form of glucose, how much of that is present within the muscle tissue as well? And then finally, how big is this overall structure? Remember, we talked about the fact that in the quads or the hamstrings, you can have hundreds of muscle fibers that are activated by one single neuron. Because remember, an axon leaves the neuron and can branch into many, many branches. So it's possible to say to have 100 muscles muscle cells acted on by a single neuron. But up in the eyes or in the fingers where you want very fine control, you're only going to have just a few, a handful, 4, 5, 6, that are activated by a single neuron. So what we're going to be talking about is we're going to go through a stepwise progression here in our description of how we develop tension. But the first thing we do in muscle is we have a twitch. A twitch is the single simplest muscle contraction there is. It's due to a single stimulus of sufficient strength that it will cause the muscle to just twitch one single time. And so there are 3 phases or 3 time periods, if you will, that account for a muscle twitch. And the first thing that we have to understand is that the electrical signals that are produced here to cause a muscle to contract, even just this one single time in a twitch, it has to be of sufficient strength to cause that muscle to actually function. And so that's called the threshold. So an electrical signal that reaches a muscle has to be a certain strength to cause that muscle to twitch. All right, and so the twitch is this single muscle contraction due to a single electrical stimulus acting on it. So there are 3 parts. The latent period is the part where the stimulus reaches the end of the axon. We go through all those steps, the release of acetylcholine, crossing the synaptic cleft, moving over to the motor end plate, causing the generation of a new action potential, it moving down over to the T-tubules and impacting on the terminal cisterns. And right when that calcium begins to get released, that's when we start the second component, the contraction phase. And that's where all that, the excitation contraction coupling happens. Then we get the actual contraction occurring. And then we're going to have a fairly sustained relaxation phase occurring. So this is the 3 minimal components for a twitch, a single muscle. All right, so this graph is correlating stimuli that are below threshold and then eventually getting stronger and stronger to how many motor nerves they'll excite, and then to what that will cause in the matter of strength of contraction. So here we see in these first 2 sets that these stimuli supplied by the brain, the central nervous system or whatever, are not strong enough to motivate or to innervate the nerves of the motor nerve that we're talking about, the actual axons of the motor nerve. This third one, though, is strong enough. It reaches threshold, and that stimulus then will excite this motor nerve and cause a very slight twitch, very low threshold. The contraction strength won't be much, but nonetheless it's for real. And in the next set, our stimulus becomes greater and we activate 2 axons in this motor nerve, and our stimulus gets a little stronger to the muscle and we get a bigger contraction. And this continues as we get more and more, and then eventually maximize our amount of nerves in this motor neuron being activated. And so the stimulus voltage keeps increasing, which increases the maximum amount of our motor nerves. But once we get the maximum amount of nerves here stimulated, we're not going to get any greater strength of contraction. That's it. That's all we're going to get. So what this shows is that contraction strength is dependent on the strength of the voltage that is being applied. Kind of makes sense. It's a direct correlation. And the strength of the voltage that's being applied correlates to the number of axons being stimulated in a motor neuron. And so you can imagine a single axon that, say, goes to 100 muscle cells leading to a certain amount of twitch. But if I activate 2, 3, 4, 5 that each go to 100, well, when I get up to 5 of these being activated, then now I'm activating 500, right? Because 5 of these axons are stimulating a group of 100 cells each, so I'm stimulating now 500 cells. Just makes sense that I'm going to have more contraction strength from that entire muscle bundle. And that's what this graph is demonstrating to you. Okay, so we have a twitch here. You can see the 3 phases, the latent phase, the contraction phase, and then the relaxation phase, the latent phase, the contraction phase, the relaxation phase, the latent phase, the contraction phase, the relaxation phase. Well, okay, so that's one twitch at a time. What happens though if we don't stimulate? You can see these little bars, these are the points of the stimulus. What happens if we begin stimulating with a slightly greater frequency? In other words, we stimulate right when we're entering deep relaxation here, right at this point. And so we start repeatedly stimulating here. And what we will see is a development of a process called TRPE. And TRPE, you can see the muscle contraction builds, but very, very slowly. And it will maximize potentially eventually if we continue this fairly slow rate of stimulus. Well, what happens if we increase the rate even more and we start stimulating before it gets down to the trough, before it gets down to the base here? We start stimulating a little more rapidly, like stimulate right when the signal gets down into this region somewhat. And that will develop what's called an incomplete tetanus. And you can see it builds much faster. The maximum contraction builds much more quickly, and as we increase the rate of stimulation. That's incomplete tetanus. And if we absolutely maximize the rate where we're pretty much stimulating at the peak, right when the contraction phase is ending, and we stimulate at that peak, we will reach a point called complete tetanus. This is actually very rare in humans. Other animals can achieve that, but humans do not, except under extraordinary circumstances. We will achieve this complete tetanus. Very rare though. And that requires an incredibly rapid amount of stimulation to occur. And it will fatigue at a certain point. All of these could eventually lead to fatigue, but it does most definitely happen during